So before I begin, one more announcement. I have moved back to my regular office, so I am now in Eucalyptus Hall on the second floor, essentially right across from the elevator, so 2226D. All right, so today we're gonna move on and we're gonna start talking about viruses. Also in the announcement that I sent out yesterday, so the material that we didn't cover last time is not gonna be on the exam. We're a little bit behind and I don't wanna take away time from the viruses because I know a lot of people were interested in viruses. So, okay. So we're going to do some basic terminology and then get into how the different classes of viruses and how we categorize them. And then talk about the different life cycles for viruses. And that we'll continue on with next time too about how we actually grow viruses in the lab. All right, so the first thing, and this is probably why it's an interesting topic to everybody. Yeah.
Sorry, so this is not going to be on the exam, right?
This one's not on Monday's exam. No. It's I forget the number, 882. The one that's like 100 yeah. All right, so viruses are everywhere. We normally think of the ones that that infect people, right? Like measles and things like that. But viruses can infect essentially all types of organisms, and so a lot of you may have heard in recent years about algal blooms. They've been making the news a lot more in recent years. So they essentially, block sunlight and deplete oxygen from other organisms. But why it's been getting into the news is because certain types also can produce toxins that are harmful to the different marine ecology. So actually, viruses. So this one is a virus that's actually infecting one part of the algal bloom. Is essentially one natural way to be able to essentially take care of the algae that are causing these problems. So these viruses will cause them to lyse and will cause the bloom to break up. Other places where viruses are actually playing an integral part is actually in you. So we've all probably heard of the human microbiome, but there's also a human virome. Some of which are actually doing good things for you, and some of them we don't necessarily know what they're doing quite yet, but people have found them. So there has been a lot of interest in viruses, and the study of viruses, and way back when some of the bacteriophage that we're going to talk about later were discovered, all of the scientists that went into that actually led the way to how we understand the basics of molecular biology and some microbiology. So things like how transcription and translation works and things like that were all helped out with the study of viruses. All right, so just some basic terminology. What is a virus? So, okay, remember way back in the first class when we were defining what is a microbe. Viruses were on the list, right? But it was under that caveat because they're not actually living, right? So they are these non-living particles. So that particle is what we call the virion. So they need to actually infect the host and use the host machinery to be able to replicate and do its thing, right? So that's why it's considered non-living. They typically have small genomes, then they can be made up of DNA, RNA, and they can be either single-stranded or double-stranded in both, either DNA or RNA. We'll talk a lot about that later. They have a protein coat, which is called a capsid, to protect it, essentially to protect its genome. And so we'll talk about this a little bit too in an examples, but viruses, they can either essentially just infect their host, use the host to make more copies of itself, and then kill the host and go on and infect another host cell. Or they can actually copy their genes into the host genomes. And like I had mentioned, there are viruses that infect all forms of living cells, and there's something about 10 to the 32 viruses on Earth. All right, so before we talk about all the stuff about viruses, I'm going to go into the caveats. Okay, so the things that kind of get lumped in with viruses, but they're actually even smaller than viruses or lack some of the general components. Okay, so the first one is the virioids. So don't get that confused with the virions. Okay, they're two completely different things. So in this case, these are actually the entire infectious particle is the RNA genome. So this here is an example of a virioid. They typically infect plants. Normally we think of RNAs as being very fragile. So they are very susceptible to being degraded. They're not very hardy molecules. But in this form, so in this double-stranded kind of looped form, this is actually very stable. And so they can actually go ahead and infect plants. They don't need the extra coat to protect it. All right, the other exception are prions. So you've probably heard of prions in the case of mad cow disease or Crutchfield-Jacob. These ones have no nucleic acids, so these are just proteins. They are proteins that actually fold into these abnormal conformations. So if this is a normal protein structure, the prions will actually fold into this structure. And then so what does that actually do? When it goes into cells, and it can actually cause normal proteins to go and misfold into these abnormal structures. And so that's kind of how it propagates, by causing the other proteins to misfold, and that can lead to all these problems. The other thing about the prions is that they can actually mutate and adapt as they go, and so that's also one of the things that makes them very dangerous. All right, so the next few slides we're going to be talking about virus structure, so the shapes and of the capsids that they make. The first couple are what we call symmetrical viruses. So the first one is icosahedral viruses. They make the icosahedral capsids, so these things here. They are essentially polyhedrons with a 20-sided polyhedron with identical triangular faces on them. Each triangle, it can be composed of 3 identical proteins. So remember now, the viruses are typically smaller, their genomes are typically smaller. So the fact that they can just use one protein to encode for their whole capsid makes it's kind of an efficiency type of a thing, and it helps with their propagation to have these smaller genomes. So for these icosahedral capsids, they have this rotational symmetry, and so the images here shows you where all the symmetrical axes are. And then some of the examples for this are some typical like animal viruses like herpes and polio. Another type of symmetrical virus are the filamentous viruses. So the filamentous capsids, so these have helical symmetry now. So you can see here, so these have identical protein units here, and they essentially just kind of go around in a helical form around, and they are essentially making this tubular shape around the genome. Okay, and so the genome of these viruses are typically single-stranded, but they can be DNA or RNA, and essentially the size or the length of the virus itself is dependent on the length of the genome, and so you'll have these protein subunits going all the way around the genome, so that way that's why it's called the helical symmetry. These are some of the examples of these types of viruses. They include phage, so ones that infect bacteria, as well as human viruses and plant viruses. Yep.
red and the blue both the virus?
The whole thing is the virus. So the whole encapsulated thing is the virus. The red is the genome. The blue is the protein code.
Oh.
All right, so another type of virus are the tailed viruses. So these ones have multi-part structures. So they have these icosahedral capsids, or in the head, what we call the head region. So that's where the genome is contained. And then they have these helical neck regions here. And then they have these tail fibers here. So an example of this is the T4 bacteriophage. And so you can see it in this image here, that they use the tail fibers to essentially find their way and bind to like the cell surface. And then they will inject so this is their genome here. This will get injected through the neck and into the cell. So then we have the asymmetrical viruses, so the ones that don't necessarily form a standard shape. These ones lack capsid symmetry. Their DNA is typically enclosed by a core envelope, and they involve many different types of accessory proteins. Some of them can also be found inside the capsid between the core envelope and some in the outer membrane. So some of them will also have these outer membranes. So some, but not all viruses can have these envelopes, so like we just discussed with the asymmetrical viruses. The envelopes themselves are lipid membranes, and so that essentially enables it for it to fuse with cells that aren't covered by a cell wall. So it'll actually be able to bind into that, and it'll get taken in by the cell because it was able to fuse using the lipid membrane. Those lipids actually, the viruses take advantage of the host, and they use the host to be able to get the lipids for their membrane. But the proteins that are in the envelope, if it has an envelope, is encoded by the virus. All right, so a little bit about the genomes. They can be any kind of nucleic acid combination that you think of. So single-stranded or double-stranded. They can be linear or circular, DNA or RNA. They are usually small, and they will encode as essentially the most necessary things. And some of the terminology here that you see for the different genes will kind of always be used to note on different viral genomes, because some of the main parts are the capsid, so it needs the proteins to code for the capsids. The envelope proteins, if they exist, and then any polymerase that it needs if it's not found in the host cell. And we'll talk about that very soon. So like I said, they tend to be smaller genomes, but there's always a caveat in all of this, and so there are some that are extremely large. So like this Mimi virus here has a genome that's essentially on the order of a bacterial-sized genome, and so it is very large. So this now encodes for other things, like other proteins, like for DNA repair, protein folding, and it has, I think, some other metabolism type genes on it as well. All right, so now how do we classify viruses? So there's this International Committee on Taxonomy of Viruses, and they came up with these different criteria just so that when people and different researchers were talking about these viruses, they can categorize them and use the same language and talk about them in these different ways. So ways to categorize them by genome composition. So we're going to talk about this a lot coming up next. The capsid symmetry, so again, things like we just discussed, like the helical symmetry, or icosahedral. their envelope, and depending on how the envelopes are constructed, size of the particle or host range. So it was thought that closely related viruses will typically infect the same types of cells. As it turns out, I think the most useful way to categorize the different viruses is based on their genome composition and how they get to mRNA. And there is greater evidence that they have a shared ancestry if you kind of categorize them by their genome types. And so this way of categorizing is called the Baltimore virus classification. So like I said, there are different types, right? Double-stranded, single-stranded, and in both cases in DNA or in RNA. So you have DNA, double-stranded DNA, single-stranded. There's also different types of single-stranded RNA, and we'll get into the details of that in a little bit, or you can have the double-stranded RNA. Okay, so now if you think about it, the virus has to go into the cell. It's, like I said, it's mostly worried about making more copies of itself. And so you have to think about how it's going to do that. Can it use the host's polymerases and machinery to do that, or does it need its own to be able to do that? Okay, so we'll go through each one of the classes and talk about them a little bit. So for the DNA viruses, whether it's double-stranded or single-stranded, it can use the host polymerase, right? So we're thinking now that they have to always make protein, so it's going to need to go through mRNA at some stage, and so the host cell always has a way to do that, right? It has ways to do transcription and translation. So it doesn't need any additional polymerases, but if you think about it in the context of animal viruses, right? So now transcription typically is going to happen in the nucleus, okay? So it needs another way to actually get its genome into the nucleus, okay? So that's an extra consideration for these two classes. So the group 1 and the group 2, if it's intended for an animal cell, it has to be able to get into the nucleus, or the other option is that it can actually, in its capsid, be carrying around a separate polymerase or genes for a separate polymerase that would allow for transcription in the cytosol. Is that clear? Sorry, which part? So the one extra consideration, so if these viruses are bacterial viruses, it's easy. DNA, whether it's single-stranded or double-stranded, the cell is going to have a polymerase that can be used directly to make mRNA. But if it has to go into an animal cell, so one that has a nucleus, the DNA now needs a way to actually get into the nucleus to be able to use the host's machinery. Okay, but not all viruses necessarily do that, and so if they haven't figured out a way to get to the nucleus, they'll just carry around a polymerase with them so that they can just do transcription translation both in the cytosol. All right, so for the RNA viruses, we'll talk about some RNA viruses next. So the first thing is the double-stranded RNA viruses. So this is group 3, so the double-stranded RNA viruses. And I'm not going to talk about the examples, but the table in your book has examples of them. All right, so now it has double-stranded RNA, and the cell doesn't know what to do that. Any cell that it's going to infect essentially can't do anything with double-stranded RNA, right? It doesn't. You need mRNA is single-stranded. You need that to be able to translate, and it doesn't necessarily have a polymerase to make more copies of RNA. It doesn't have a way to do that, and so what it needs are what's called RNA dependent RNA polymerase. Okay, so the RNA dependent part, meaning it's going to read RNA, and then the RNA for the polymerase part, meaning it's going to make RNA. So it needs this, and typically what's going to happen is that this is going to get, in the case for these group 3, they're going to also just package that actual polymerase inside the capsid. So as soon as it gets into the cell, it's going to have this polymerase, so then it can go off and start copying its genome, getting to the mRNA that the cell can actually read. And then in that genome will be sequences to make more of this, so that when it's packaging itself up again before it exits the cell, it'll be able to put in a polymerase with its genome. All right, so group 4. So you want to pay attention to this, right? So these are the plus sense single-stranded RNA. So now there are 2 ways of being single-stranded RNA. So the plus sense, so this can serve directly as mRNA, right? But so this can go ahead and can translate and can make proteins from this, but this still needs a way to make more of its genome, right? Because it's going to make the proteins, but it still needs to copy the RNA genome. So it's, in this case, still needs that RNA-dependent RNA polymerase, so this RDRP. So they still need that to make copies of its genome, but can be used directly to make proteins from this. Yep.
Do we need to remember the group numbers and what goes with what, or just what each one does?
More so just what each one does. Like, what does each one need?
Sorry, when you say a virus needs something, do you mean that they have that machinery within them, or do they seek that from the host?
So when I say needs, I mean it needs it to be able to replicate and to essentially lyse the cell, do its whole job of like trying to make more copies of itself and get out of the cell. So it can do that by getting it from the host, or it can bring it in on its own. So with these, yep. All right, so now we have the minus sense single-stranded RNA. So now this has to go from minus RNA to plus the mRNA version, essentially, before it can start making proteins. And it also needs to copy, so this one also needs an RNA-dependent RNA polymerase. Essentially, all the RNA viruses will need a polymerase. But it's important to realize that it has to go through this extra step for the minus strand RNA viruses. All right, so now we have retroviruses. So these are typically single-stranded RNA, but it goes through the single-stranded DNA intermediate before it gets to double-stranded DNA. And so in order to go from RNA to DNA, it uses a reverse transcriptase. I'll often call this RT. So the reverse transcriptase can actually do both of these steps. So it will take a single-stranded RNA, make it first to single-stranded DNA, and then it can also do single-stranded DNA to double-stranded DNA. And then these typically, the double-stranded DNA will get integrated into the genome, into the host genome. All All right, so the last group are this pararetroviruses. So these ones go from double-stranded DNA, and then it goes to RNA, and then it gets reverse transcribed back to DNA. These ones are some of the types of viruses in this class, they infect plants, and so some plants actually have a reverse transcriptase that can be used for this, but otherwise, this is something that the virus is going to have to provide. All right, does the classifications make sense, the genomes and what types of genomes and what extra requirements that now brings for each virus? Okay. All right, so now we'll talk about viral life cycles. And in general, all viruses have to go through this process, okay? And we're gonna only focus on the bacterial viruses, so bacteriophages, and we're gonna go through each of these steps for those. So initially it will start with host recognition, so it needs to, the virus needs to be able to find the host. And in a lot of cases, right, these viruses, they're very specific to specific kind of host cell, so they have to have a way to recognize the host that they're interested in. Then they have to get their genome into the cell. They have to figure out ways to replicate their genome. They're going to spend some time making all the viral proteins and then finally assembling, again, more viral particles, and then a way to exit and to spread into more host cells. Okay, so for bacteriophage, these are viruses that infect bacteria. For the first part, so the host recognition and attachment. So they will always find these cell surface receptors on the surface of cells, and it will most likely be, so like in this picture, it's always going to be something that is required for that cell. So it's something that the cell is using all the time. And again, this is going to be specific to the bacteria. There are phages that are very specific to species, and so they are going to be based on whatever surface receptor that they are going to be attaching to. The second step is the genome entry. So most of the phage, they will inject their genome only. So it's easiest to imagine with the tailed viruses, right? That whole image of they've now found the receptors and they're literally injecting their genomes into the cells. And then a lot of times what will happen is they're going to inject their genomes in here, but their capsids will still remain on the outside, and so they call those ghost capsids, so they're empty.
So in the last slide you talked about the receptors.
Mm-hmm.
They just use the receptor to say, hey, that's the cell I'm looking for, not as an entryway, right? Just to identify it.
Sometimes it is an entryway. It depends on the virus, but it's definitely the first case is to find the cell that it's looking for. All right, so for the last few steps, okay, so how it's going to replicate its genome, make the viral proteins, assemble the capsids, and exit. So this is where, when we talk about bacteriophage, we have different types of viruses. So we have things that can do the lytic versus the lysogenic cycle. And so just to make it a little bit more clear, these are different types of viruses. So you'll have viruses that can only do lytic, and then there are viruses that have this option of doing lysogenic before they go into the lytic cycle. Okay, so first talking about the lytic cycle. So the main goal for these types of viruses is they're going to use their host to make as many copies of themselves as possible and then go out and infect more cells. So what it's going to do is essentially, you know, replicate everything, make all the proteins that it needs, assemble new phage particles, right? But now it has to kind of be smart about it. So some of the genes that are required to do this are so it needs to make the protein capsid, right? It needs to copy its genome, and then in order to exit, what it actually does is it usually produces some kind of enzyme that is going to help it get out of the host. It's going to produce some kind of enzyme that's going to lyse the host cell so that it can get out. But if it were to just go in and just make all these things just right away, now the host cell is going to start lysing and it hasn't actually finished using the cell to make more copies and didn't give it enough time to package itself up, right? So what they've done is they've evolved ways to essentially have these early genes where at first all they're doing is making copies of the genome and then making the capsids. And once it hits a certain concentration of that, then it can go ahead and produce the late genes where it's going to start producing the enzymes to lyse the host cell. So that way it's timed it out perfectly so that it can get maximum efficiency. And so this lysis is referred to as burst, and the number of particles released is called the burst size. We'll bring this up a little bit more when we talk about how we grow phage in the lab. All right, so for the lysogenic cycle, now another name for this type of phage are temperate phage. So now this one's, they are now have the option of integrating their genome into the host. So once it's integrated into the host, it's called a prophage. And so the other kind of advantage of the prophage, so now once it's in the genome and now it essentially like stakes its claim on that cell. So other viruses that are able to do this can actually go and integrate their genomes. So what happens is it goes in and it gets into the host genome, and then the host just kind of goes along and does its thing. And so it gets replicated along with the host genome. And then eventually it will reactivate and it will head towards the lytic cycle. So it's going to go ahead and excise itself out of the host genome, and then it's going to go through and replicate and make more copies and then lyse the cell and get out. So usually what will happen in this case is that some kind of environmental stress will trigger this. So the virus, again, you know, its main goal is to survive, replicate, make more copies, go infect another host. So if it's sensing that the cell is going to die, it's going to say it's time to get the hell out of here, and it's going to get out and go and hopefully find new host cells. But other times this will actually just occur at random, and we haven't quite figured out how that works. One thing that's going to come up later in the class is, so while the viruses are exiting from the lysogenic state, and so as it's excising, it can actually accidentally take out part of the host cell genome. And so when it does that, it gets packaged up, it packages up some host DNA, and then when it goes into another host, it's going to go put that in there. And so this is one way that cells get other DNA from other cells. All right, so this is just the pictorial overview, and we will watch the video about the cycle. But this here is essentially the lytic cycle, right? So you have the phage infecting the cell, the genome's in there, and it's going to go and it's going to make all these particles. It's starting to, all the proteins. Now it's starting to assemble the different, the capsids and get the genomes in there, and then it will lyse the cell and get out and go to new places. This is now versus a lysogenic phage that's going to go in, and the first thing it's going to do is integrate itself into the host genome. And then as the host replicates, so this now it goes into all the new cells. But at some point, when it's time, it will actually go and excise itself from there, and essentially it will slot back into this part of the lytic cycle. But again, don't let this figure confuse you. These are two different types of viruses, okay? So there are viruses that are only lytic. Not all viruses have the ability to integrate into the genome. Okay, we'll just watch this video as a recap of what we just talked about.
Unlike a virulent phage, a temperate phage, such as phage lambda, does not necessarily cause lysis after entering a host cell. Instead, the temperate phage may embark on a pathway called lysogeny. In lysogeny, the phage first attaches to its host and injects its genetic material inside the bacterial cell. Linear double-stranded DNA is then circularized. The circularized genome then recombines into that of the host by site-specific recombination of DNA. The integrated phage genome is called a prophage. Lysogeny is a condition in which the phage genome is replicated along with that of the host cell as the host reproduces. Implicit in the term lysogeny is the ability of such a strain to generate a lytic burst of phage. The regulatory switch between lysis and lysogeny responds to environmental cues. That is, how likely is it that the host cell will survive and continue to propagate the phage genome under present environmental conditions? If the cell's growth is strong, it is more likely that the phage DNA will remain inactive, whereas events that threaten host survival will trigger a lytic burst. At the beginning of the lytic pathway, the phage genome directs its own excision from the host genome. As it exits, it circularizes again. A viral DNase cleaves the host cell's DNA. Using information in the viral genome, the cell synthesizes capsid proteins. The cell replicates the lambda phage DNA by a rolling circle method, making many copies that become packaged into the capsids. A phage enzyme then lyses the cell, and the progeny phage spill out, ready to infect another host cell.
Any questions about lysis and lysogeny? Yep.
How are the viruses mobile? Are they just, because they're like dead, right? Do they just happen to fall where they need to? Or like, are they actually mobile and can?
So if you think about it, usually it's in some kind of cells are usually in some kind of aqueous environment. It's also just they move by diffusion. So they're not necessarily, well, some of them might, but you don't usually think of them as like swimming. But they can move around just with the movement of the media or the environment that they're growing in. All right, so one more type of phage life cycle. So we have lytic, lysogenic, and now we also have the slow release cycle. So essentially these ones now, they're gonna go in and instead of killing the host, it's just gonna slowly just like make one copy at a time of itself, and then it has a way for it to exit the cell. So it's just gonna use the host to slowly make one new virus particle at a time and continually use the host to do that. So it's not going to make tons of particles and kill the cell. It wants to keep that host around and use it as long as possible. All right, so let's see what you guys think about what are some of the advantages and disadvantages from the perspective of the virus of slow release versus alternating between lysis and lysogeny. So what might be reasons why you would be like a temperate phage would be better, or why might the slow release be better. Yep.
Just depending on the host, it could be more fitting for either way. It's like a slow release of a cell that's more stable or something, or lysogeny for something that's in a less stable environment or something.
You mean the host cell is in a less stable environment? Okay. Yep.
For viruses in the lytic cycle, they're most likely not able to. They're not able to go in in other cycles, so that they also need to, reproduce quickly, or they would need a.
So we're talking about the lysogenic versus the slow release. So the ones that can go between.
Okay.
Yeah, not lytic on its own.
Okay. Cool. I would think that maybe for slow release, they wouldn't want to probably kill the cells because they still want to take advantage of the cells.
Mm-hmm.
So they want to keep them alive and without killing them. Whereas
So why would that be an advantage?
because it's it's almost like unlimited resources.
Okay. Maybe it'll survive longer, right?
Uh-huh.
Yep.
Slow release maybe would be better on the long run, but if it's like urgent and the virus want to get out, so it will use the method of lysis and lysogen.
Yeah. So
Copy and run it.
right. So in the case, right, the slow release, there's no choice. It can only go a little bit at a time, right? So it's never going to make as many particles in a certain amount of time as one that can do lysis. Yeah.
I hear what you said, but does it depend on the host?
So what about the host might it depend on?
Characteristics, how it behaves.
Maybe. What kind of behavior? Or somebody else can answer. Okay. Yeah.
Depend on how fast it doubles, how fast it reproduces.
Yep. So it can depend on how fast the host is reproducing. So what other things related to host replication might be a part of it? Go ahead.
For slow, it depends on the immune response of the host cell. So the slow release allows for the cell to the host cell to have more time to protect itself, maybe.
That's a good one, right? So if it's slowly using the host machinery, if the host has some kind of defense mechanism, that's going to give the host time to fight back. So that's a good one. I saw somebody else raise their hand up here. Yes.
Would it not depend on the environmental conditions, like whether it's favorable or unfavorable?
Yep, exactly. So if, let's say, the host cell is in a bad place, right? Being able to quickly switch to lysis and make to the lytic stage and make lots and lots of copies of itself, that's a good thing to have, right? Versus, again, if it's a slow release one, if the cell, all of a sudden it looks like your host cell is gonna die, it can't go and make any more copies any faster. It's just, this is the one speed that it goes. So it can definitely depend on the environment of the host. Yep.
Slow release also related to the adaptive immunity and temperate immunity, or
It could be. I'm not sure which one would be more advantageous over the other, but yeah, it would definitely relate to immunity. Yep.
can we say it's more advantage or disadvantage according to the kind of viruses that we have?
What do you mean?
Different viruses can have different lifespan or different effects on the host. So it can be an advantage to a kind of viruses, but a disadvantage to another.
Yeah, and that's going to have to do a lot with the host, right? And what's happening with the host and where it's living, if it's counter-attacking it, things like that, all things that we've talked about. Yep.
For the slow release, it would be advantageous to be able to keep the bacteria alive longer, because if the bacteria is mobile and has sensing proteins, it could move you into an area that is advantageous.
Yep.
And continue to slowly release the virus into a densely populated
Yep.
area.
Yep, that's a good one. Anybody else? So one more thing about the slow release viruses. So they're actually taxing the cell. So they're using up the cell's energy and resources, and so that can actually cause the cell to essentially grow slower. So it's going to use up some of the resources, and so the cell is not going to divide as much, and so that is one disadvantage, that it's going to lead to essentially less particles being released because it's going to be dividing slower. All right, good. So last thing, kind of got brought up, but bacteria have developed ways to defend themselves against viruses. They can form they can evolve genetic resistance, right? So the phage need to recognize the host receptors, right? So they can evolve a mutation in that, and now all of a sudden the phage can't actually find it and bind. There are restriction endonucleases that will cleave the viral DNA, so the cells will recognize that the viral DNA is different than their host DNA because of this methylation, which we'll talk about actually next time. But so it has ways to recognize whether it's a foreign DNA, and so it'll cleave that. And then a lot of you have probably heard about CRISPR. It makes big news because of what we can use it as for engineering tools, but its original use was a bacterial immune system. So again, it's going to find the virus, recognize it, it's going to store it, and then now when the host actually gets infected again, by that same virus, it'll recognize that, so it'll attack the virus right away and save itself from it. All right, that's it.